I will talk today about our studies of the effects of radiation on human tissues and organs and development of countermeasures for long-term space missions. The first step in this project was to assemble the team, and we really gathered the experts in all areas that we believe are relevant for this kind of research, from radiation physics and radiation biology to tissue engineering, nanotechnology, drug testing, pathology, modeling and statistics, and finally systems biology. Our goal was and remains to be to study the effects of space radiation and to develop countermeasures for prolonged deep space missions using a novel technology that is called organs on a chip platforms. The funding support for this effort is provided by the Translational Research Institute for Space Health. In addition to this immediate goal, we were also thinking when developing the project about how to also have impact on space in a broader space research, in a broader sense, and also impact on population on Earth. We believe that these studies can help both evaluate and mitigate the risks that astronauts may encounter in the space mission, and that we can do it in an individualized manner, taking into account physiological differences between individuals. Second impact is that these studies will help to some extent to understand the effects of acute and also chronic exposure to simulated space radiation, and at the same time help us evaluate different types of radiation countermeasures and to develop countermeasures that would be really suitable for use in space. Impact on Earth is that these studies will help assess efficacy of radioprotective agents that can be helpful for people who are exposed to radiation clinically or as a part of their job, and also to better understand the fundamental basis for the immediate and long-term effects of radiation on humans. This is our plan. To this end, we developed these three specific aims. Aim one is really to identify the changes that happen following exposure to radiation in multiple organ systems in the human body. To this end, we started from iPS cells, induced pluripotent stem cells that are derived from small samples of blood, and we engineered and matured human tissues that are of interest to these studies, and I'll tell you a little bit more about why we selected liver and heart and bone marrow and vasculature. We would assemble a tissue platform with these individual tissue systems, and we would study how they respond to either acute or chronic dose of radiation with different types of radiation and different regimes of exposure. The effects were measured at the tissue level, both structurally and functionally, and also at the cellular and subcellular level, looking into the secretion of cytokines, into the changes in cell phenotypes, and into results of sequencing. Aim 2 is pursued in conjunction with Aim 1 in an integrated fashion. So this aim is about developing countermeasures. And we started from using an agent that has been shown clinically to be very effective to protect patients from excessive radiation. So this is GCSF, granulocyte colony stimulating factor, which is very effective and taken in a free form, in form of injection, something that's obviously not suitable for use in space. So we proposed to develop a nanoparticle-based system for long-term sustained release of GCSF that can be taken periodically orally as a pill and get into the human system. An alternative measure that we are also looking into is to augment the ability of the body to produce this molecule and thereby provide a kind of intrinsic protection of the tissues exposed to radiation, even without taking the medication. The evaluation measures are exactly the same as the ones that I mentioned in AIM-1. And AIM-3 is where everything comes together, and we have two related goals. One is to generate platforms that are individualized. So we would really like to generate astronaut on a chip platform and enable NASA to test in advance for each individual that we will be flying to space, their response to radiation and their response to the tissue protection measures. This will allow really optimization of the protocol for each individual. The other goal is to do these studies in the Brookhaven National Lab, which has the absolutely best simulated space radiation system that we can use for the exposure of our tissues. So let me tell you a little bit about organs on a chip, about this platform that we are using in the studies. I mentioned that you start from an individual. You will take very small test tube of blood, derive stem cells. You will use these stem cells to generate different types of tissues. You will engineer them and mature individually. And then once they are brought to the stage when they're resembling the adult tissue functionality and phenotypes, you would combine them into the platform. So you see this in this little video underneath. For this platform, there are quite a few design criteria that you really need to follow in order for these platforms to have utility for both research and translation. One is that they need to be configurable so that you can select the tissues that you're interested in to form specific physiological units that will depend on the questions that you're studying, and also allow you to vary the numbers of individual tissues and the types of the tissues in the order in which they are connected. Biological specificity is very important. This is provided by the use of iPS cells so that you can really do studies for a specific genetics and other conditions. And also this allows you to parse out the effects of genetics from those of environmental conditions. The very difficult requirement is to have stable and mature tissue phenotypes that recapitulate the adult human situation, and you need to maintain these mature phenotypes for weeks to months for the platform to be useful. At the same time, you need to allow the tissues to be connected by vascular perfusion so that you get physiologically meaningful conditions. And finally, we are relying as much as possible on real-time readouts so that you can do studies, you can do dynamic studies, and you can also do longitudinal studies. So you look over and over into the same tissue sample. So how do we do that? You can see here that we are in this in one particular case, we assembled the liver tissue, heart tissue, bone tissue, and skin tissue. All are derived from the populations of cells differentiated from iPS cells. They're matured in culture and they're combined in this platform, as I showed you on the previous image. So the main design feature of this integrated platform that we are using is that between the tissues and the vascular perfusion connecting them is endothelial barrier. So we are really combining these conflicting requirements that tissues need to be stable with the requirement that they need to communicate with each other, need to be connected with each other, but just borrowing from the recipes that our body is using. We are all about compartments and boundaries. So we are putting this endothelial barrier, a boundary between the tissues and the vascular perfusion. And this barrier is selectively permeable both to molecules and to the cells. So one nice example that you see here at the right is that we showed that if we circulate immune cells in the system, in the vascular circulation, then they would just stay in the circulation. However, if one of the tissues is injured, then the trophic factors related to injury are mobilizing the endothelial barrier opening and letting the immune cells to get into the tissue. You can see this here at the far right, where the immune cells are labeled and they really show as infiltrating the tissue. So using tissue engineering methodologies, we engineered a number of different human tissues that I show here, and you never use all of them. You will really just use a subset that is important for the question that you're studying. So in this particular case, in this project, we are using a liver tissue, which is pretty much obligatory component of all our organs on a chip platform because it's a site of metabolism. We are studying the heart muscle, which is known to be the target for delayed or chronic radiation damage. And we are studying bone marrow, which is a target of immediate or acute radiation damage. And then, as I mentioned, we are connecting these tissues by circulation, and there is a vascular endothelial barrier, which we also need to have. And then we can have circulating immune cells in the systems because they're very important. They always are a big player in any condition of injury or disease. So let me just show a little bit of details so you can see what are the tissues we are talking about. To engineer bone marrow, we start from the bone tissue that we form from the mineralized bone matrix that we populate with the component cells. So these are iPS-derived osteoblasts supporting mesenchymal stromal cells, endothelial cells, and then we also add the CD34 positive hematopoietic cells. So after a period of time, it takes 4 to 6 weeks, depending on the donor of cells, we obtain a bone marrow module. And you can see here at the bottom that this bone marrow is really expressing all markers that you would like to see. We benchmark these tissues against their native counterparts. On the top right, you see that bone marrow has, the cells in the bone marrow have a colony forming ability, which is critical determinant of bone marrow, and then also both the CD34 positive and CD45 positive cells are proliferating, so there is a hematopoietic expansion. When you subjected this bone marrow module to the platform, to the radiation, you see here that there are structural changes. So what we compare here in the first column is naive tissue not exposed to radiation, tissue exposed to photon radiation, and tissue exposed to neutron radiation. And what you see here at the right is that in response to radiation, just an example of many functional data that we collected, in response to radiation, the cell count for CD45 positive cells, the same thing you see for CD34 positive cells, the cell count goes down. However, if you irradiate the tissues in the presence of GCSF, they are protected from the harmful effects of radiation, at least in this acute setting that we studied. The other tissue of interest is the human heart muscle. Again, we formed this muscle that you see here at the top left, beating very nicely in response to electrical signals that are designed to mimic those in our heart. The muscle has a very nice aligned striated structure that you see at the bottom left. A little bit more insight into detailed structure shows you that there are registers of sarcomeres. This is the contractile apparatus of the cells with mitochondria. These are these dark blobs here that are aligned and positioned along the sarcomere. This is the energy factory of the cell. And this high density of mitochondria that you see here at the bottom right is physiological, and it is associated with the switch to oxidative metabolism that cells do in order to effectively utilize nutrients and oxygen. At the top right, you see cross-section through the muscle that reveals very nice morphological features that underlie the functionality of this muscle. So when we irradiate the muscle, data are shown here just for neutron radiation studies. You see that much of this nice striated structure is lost. And when you look into the functional data, you see that they are that the tissue is degrading its functionality when exposed to radiation, but not if the radiation is applied in the presence of radiation protection agent. So these are very promising initial results that we are working on in a very detailed fashion in an attempt to really learn more about the underlying biology and also optimize any countermeasures that we may be able to develop. So in summary, what I try to tell you today is that we have methodologies to engineer matured adult-like human tissues of interest, starting from iPS cells. We can connect these tissues by vascular perfusion, and under these conditions, we can maintain their phenotypes in culture for weeks to months to support these studies. Although these tissues are very small and they're very simple compared to the native tissues, they still have ability to mimic the human physiology at the organ level. And this actually explains the name organs on a chip. So this is what they mimic rather than what they actually are. Radiation damage and radiation protection can be studied both at cellular and tissue levels using these systems. So we are doing these studies in individualized settings using iPS cell-derived tissues. We have ability to expose tissues to both acute and chronic regimes of radiation that actually simulates the actual cosmic radiation. We are working on the developmental testing of different types of countermeasures and on identification of genes and pathways that are involved in radiation protection. What is really nice about this project is that there is a cluster of several projects done in several laboratories, and we are communicating with each other and just trying collectively to make this critically important advance, both for NASA and for certain clinical benefits on Earth. And at the end, let me just thank my lab. So this is one of the recent photos when we finally had a night out after a very, very long time under COVID. I would like to acknowledge our funding sources, and a big thank you goes to our many collaborators that are instrumental for everything we do. So thanks again, and if time permits, I would be very happy to answer any questions that you may have.